So our next talk is going to be Dan Oliver. He's a co-founder and CEO of Rejuvenate Bio, which originated from George Church's lab and is focused on genetic diseases in both animals and humans. So please welcome Dan Oliver as he delivers his talk, Silencing the Critics, Scaling Gene Therapy for Human and Animal Health.
Awesome, thank you. Yeah, so first couple of slides here I can just kind of jump into, basically summarize kind of key critiques of gene therapy that I think a lot of us here I don't think are inherent to the technology, and then there are ways of solving these things. So we're Rejuvenate Bio. We primarily focus on, if we could advance, primarily focus on gene therapies to treat polygenic diseases, particularly focused on aging. And you'll see that play out here in a number of different facts. But I think we've all heard these critiques, too expensive, can only focus on niche applications, and there's like dramatic manufacturing struggles. And now layer on top of this, that's an inherently unsafe technology here going forward. And so just wanted to provide a different perspective on ways we've addressed all three of these things to make scalable, safe, and actually cost-effective gene therapies that should allow us to get these to literally millions of patients. And so first off, I'll talk about marrying delivery targets and manufacturing yields to create scalable gene therapy solutions. Actually validating our platform and our therapies such they are so inexpensive in the animal health market, so to treat companion animals like dogs and cats. We've actually been able to sign a number of strategic partnerships here, and actually should be commercial on this as we're moving into human clinical trials. So offers an ability to actually generate revenue in a much faster way, and hopefully, solve some of these capital efficiency problems with gene therapy. And then I'll tell you a little bit about, not just the target selection and how we deliver, but on the actual manufacturing platform that has also helped driven the COGS of this to such low levels that it's relevant for animal health. So just to start with our first human program, we are interested in delivering a gene called FGF21. We do this via a liver-tropic gene therapy with liver-specific promoters. It's AAV8, and we think this is another target that has an opportunity to have a GLP-1-like profile, where you're not just targeting on a very specific cardiometabolic disease, but have the ability to expand out into multiple different diseases. Now, saying that, I think the really interesting thing is we actually have developed really interesting data in very specific niche applications for this technology. And so this is our gene therapy, again, delivered primarily it's delivered via one-time intravenous injection, primarily is infecting liver tissue and expressing kind of our key protein from there. But we're actually outcompeting monogenic gene replacement approaches that are cardiac-centric for a monogenic disease. So this is a PKP2 knockout model. It's a particular type of arrhythmogenic cardiomyopathy. There's actually multiple people going after PKP2. This is the gold standard animal model. We actually reduce arrhythmias in these animals' hearts. Actually, we do a better job of reducing them, and we do it at a far lower dose, even though we are not directly targeting cardiac tissue on this. And so even in a kind of rare disease context, we are having superior effects here. A number of these folks are already in human clinical trials for PKP2. We'll be moving forward actually initially into a subpopulation that they cannot treat. There's a variant called desmoplakin, or DSP. Unfortunately, the DSP gene is too large to be packaged directly into an AAV, and so we can start in a kind of non-relevant genetic population for our competitors, but also generate data that should be relevant not only for the PKP2 variant, but all of the kind of secondary and tertiary genetic variants out there as well. So this is a way of actually getting to more patients with lower N values here as well, with a more broad-based therapy. But again, it shouldn't just stop here. There's actually been 3 major transactions for FGF21 protein analogs that are going after things like MASH, and then hopefully going after other cardiometabolic diseases. So excited there on that. All right, and so probably the thing that makes us the most novel, although there have been at least one other gene therapy company that's gone after animal health, but we have driven the cost of these therapies to the point that we've signed now 5 corporate partnerships with the leading animal health pharmaceutical companies. So this is a much smaller world, but in some sense that makes it easier. I basically know all the folks that we need to know in the animal health world, but basically we have a program in osteoarthritis, a program in leading type of heart failure, and then follow-on programs that should be able to basically make long-acting versions of almost any biologic the animal health industry is in. To give a perspective of how large these things can be, there's recently been a lot of success in monoclonal antibody therapeutics in animal health, and multiple products are now selling close to $1 billion per year. And so this is big money. This is not like a backwater industry. The largest company, I think, has a $90 billion market cap. And so this is a very viable path to moving these things forward. And the regulatory framework allows you to effectively commercialize these in the time frame it would have taken you to do IND-enabling studies for human health, is kind of the way of generally thinking about it. So this is mitral valve disease. This is the lead program we're going after. It's the leading type of heart failure in dogs. There's about 90 million dogs in the United States. Mitral valve disease afflicts about 7% of them, so about 7-ish million dogs, something like this. Poster child dog is the Cavalier King Charles Spaniel. I bring this up because there is kind of a corollary for the rare disease treatment groups that we deal with in the arrhythmogenic cardiomyopathy side. Is I've actually been the keynote speaker at the American Cavalier King Charles Spaniel National Club the last 4 years. Their owners / we call them pet parents are just as passionate, and unfortunately, for these folks, they have experienced their dogs getting this same disease and dying it sometimes 2, 3, 4 times. And so are quite knowledgeable, quite passionate about finding a solution for this, and have actually put up quite a bit of money to help us move this program forward as well. So let me see here. So just a little bit of the data that we've been able to demonstrate here. These are dogs that have mitral valve disease. They are segmented into different progressions of that disease. We intervened at a particular time point called B2. We did it in conjunction with the current standard of care, which is an inodilator that helps reduce the load on the heart. And you can see we basically were able to give these dogs approximately 2 years, a little under 2 years of extended life here. I always like to tell people, like, the dog years thing actually does kind of work even in heart failure progression. And so you're talking about something that would be akin to 10-ish to 14 years of progression in humans on this side for the dogs. And just a little bit about the manufacturing platform that has really helped us get over the finish line with reducing the cost of this. We're utilizing the WuXi Advanced Therapies, and now called Minaris TESSA system. And so instead of utilizing triple transfection, which we experiment with and got our cost of goods sold somewhere in like the $600 to $700 per dose range, the TESSA system is driving us below $100 per dose. The kind of key innovation with the TESSA system is they utilize 2 different adenoviruses to deliver your 3 pieces of DNA. So instead of utilizing a whole batch of GMP quality DNA for your capsid, your gene of interest, and the helper protein, the adenovirus provide the helper, the helper proteins or genes there, and then your GOI and your rep cap are actually delivered via the adenovirus, and they have great ways of clearing that. Going forward, we're actually pushing this. We'll be the first to take this through GMP qualification. The Center for Veterinary Medicine at the FDA actually relies on CBER for their CMC qualification. So we'll have this qualified in the same way you would for a BLA on the human side. It'll just happen faster on the animal health side. And so we hope to be able to prove this out both for our partner Minaris, but also for ourselves, that this is a viable path with the technology dramatically increases yields about 10x and actually reduces input costs as well. For this as well. So again, another proof point that this isn't inherently costly from a COGS standpoint. I think there still are a lot of questions on kind of one-time costs that can build up for small patient populations, but I think this actually absolutely frees us up to look at very large patient populations and then opens up gene targets that hopefully could span the gap between both. So happy to talk about that with everybody. Yeah, thank you.